So, we are now going to talk about the small intestine. So, if you are following along, we've talked about the oral cavity, the esophagus, and we've talked about the stomach. So, the stomach ends with the pyloric sphincter, and the duodenum begins with the pyloric sphincter. The pyloric sphincter kind of acts like a door. When it's open, food contents called chyme move into the duodenum. The duodenum is interesting because it is the shortest section of the small intestine, and it is still responsible for breaking down food. The rest of the small intestine does not have that function. The duodenum is the only one that still breaks down food. And then we have the jejunum. Jejunum means empty. That's what the Greek stands for. And when you cut into the jejunum, it will actually be empty. Its function is to absorb carbohydrates and proteins. And there's a whole membranous structure back behind here called the mesentery that's responsible for doing that. Then we have the ileum, which is the last section of the small intestine, and it ends at the ileocecal valve, which then goes into the large intestine. It's responsible for absorbing B12 and bile salts. And you may be wondering what bile salts are. Well, I'm going to talk about those when we talk about the liver. Now, the inside of the small intestine consists of all these little folds called villi. And if you look here, you can see it's folded, and then on top of those folds, we have these little villi structures. And you may or may not be able to see this when you cut into your small intestine. We'll look for it, but like I said, you may not see it depending on your cat. Now, really important about villi. Villi do not move food. A lot of people have this misconception that villi kind of wave. I think because they confuse them with cilia, but they do not move food. They don't wave. They don't in any way move at all. What they do is they create more folds, which allow for greater surface area along the small intestine. And that's really important when your job is to absorb stuff. So you want as much surface area as possible. Now, what does move food through the small intestine and all of the alimentary canal is a muscle contraction called peristalsis. Peristalsis is a muscular contraction. It acts in kind of a wave-like motion where you squeeze the top end, pushing the food down, and then it slowly closes and opens, pushing that food throughout the alimentary canal. That is called peristalsis, and that really only happens to push food. Villi does not wave or in any way move food through the small intestine. Now, the lovely parasite we're going to be talking about during the small intestine is the tapeworm. Diplydium caninum is the tapeworm that we'll be looking for in the cat. We may not see any. I have yet to find a tapeworm inside one of our cats, but it could be there. And so I want to talk a little bit about tapeworms. Now, the one that infects humans the most is beef tapeworm. And this is actually a picture of beef tapeworm. And I want you to realize this little white line down here is actually a foot-long ruler. So you can imagine just how long this beef tapeworm is. There are two structures that make up a tapeworm. One, there is only one of, the scolex. There's only one scolex on a tapeworm. The rest of the tapeworm is made of little structures called proglottids. And the way the proglottids look actually gives tapeworm its name. And so let me talk about that a little bit. First, the scolex. The scolex is a structure designed only for attachment. This is not a head. There is no nervous system. There's no brain. There's no mouth. A tapeworm does not have a head. It has a scolex. And the scolex's only function is to attach in between the villi of the small intestine. Sometimes they have a series of hooks. Sometimes they have a series of suckers. Sometimes they have little spikes. In fact, the scolex, the way it is shaped, is specific to each species of tapeworm. That is how we are starting to tell them apart by looking at their scolexes. Now the proglottid. The proglottid is the little segment. And if you look at our Diplodium caninum, you can see the proglottids here have broken off. The proglottids are what we call those little segments of the tapeworm. Inside of the proglottid, there are male and female reproductive organs. And oftentimes a tapeworm will just self-fertilize. All of these little dots right here are eggs. There can be thousands of eggs inside of a tapeworm. Now, when a proglottid gets fertilized and it has a whole bunch of eggs in it, that proglottid is going to break off of the rest of the tapeworm, and it is going to either break inside of the host or it's going to be passed entirely out of the host, and then it will break open, releasing the eggs. Understand that the way that tapeworms get nutrition is through absorbing materials through their epidermis. So they absorb the already digested materials through their skin. That's how they nourish themselves. So they don't really even have digestive organs. They have a little bit of things to help their cells function, but otherwise not really anything. Most of what their life cycle is devoted to is producing as many eggs as possible. So here we have tapeworm, Diplodium caninum. The scolex embeds in between the villi. It hooks right in there, and then it starts growing. It starts producing more and more proglottids, and those proglottids self-fertilize and then break off, and the eggs get released with the feces. Those are then picked up by the larvae of fleas. They eat the eggs for nutrition. But instead of them actually being digested, they just grow inside of the flea. And when the cat is grooming itself and eats these fleas, then it re-ingests all of those little larvae tapeworms, and it gets another infection of Diplodium caninum. Now, accessory organs, the liver and gallbladder. Here's the liver. This little picture you can see, here's the gallbladder. The liver is responsible for secreting bile. So here's the liver, here's the gallbladder, this little brown thing. Now, the liver produces bile, which helps to break down lipids. It leaves by the bile duct and enters the duodenum. Remember how the duodenum helped to digest food? Well, it doesn't have any way to make those digestive juices, so it relies on some other glands to help make them, the liver being one of them. The gallbladder, on the other hand, stores any excess bile that the liver produces. So you make bile in the liver, any excess is stored inside of the gallbladder. The pancreas also secretes digestive juices and puts that into the duodenum. So I love this picture. You can see the liver and the gallbladder entering the duodenum, and you can see the pancreas. Here are the digestive juices produced by the pancreas. Also insulin, you may have heard of before. That is also a digestive juice. And that's where I'll end.